Hello, good morning everyone. So today I am going to present about the forest above ground biomass estimation using the polyometric SAR. And this is the brief introduction about the importance of the forest. So this is the brief importance about the forest. As we know that forest covers around 30% of the global land surface, and they contribute around 45% of the terrestrial carbon. And most of this carbon is stored in the form of the above ground biomass. And particularly the deforestation problem is happening in the tropical forest. So there is a need to estimate the more accurate forest above ground biomass in the tropical forest. And this research is also related to the United Nations Sustainable Development Goals. And how we can estimate the forest biomass. They are different. Okay. So how we can estimate the forest above ground biomass? There are different techniques. First is we can estimate using the in situ field survey data, but it don't give you the regional dynamics of the forest biomass, and it's very difficult to conduct the forest field survey in the harsh mountain regions and in the wild intact region of the forest. And then after that, the remote sensing comes into the pictures, and there are different remote sensing platforms that can be used to estimate the forest biomass. First one is the optical remote sensing, but as we know that the optical remote sensing data is affected by cloud cover in the tropical region, and the another limitation is that it cannot penetrate through the canopy of the forest. The second one is the LiDAR data, and LiDAR data is very costly to conduct the survey in the tropical forest, and they are mostly the developing countries. They cannot afford the frequent LiDAR UAV survey in the tropical forest. And the fourth one is the microwave remote sensing SAR data. This microwave remote sensing operates in the microwave region of the electromagnetic spectrum, that is -1 to 1 millimeter, and it can penetrate through the cloud cover, and it's not affected by the weather condition, and it can operate day and time. So, but using the SAR synthetic aperture radar microwave data also, there is limitation that the subcanopy conditions in the forest is still poorly understood. So this research, and using the synthetic aperture radar, most of the algorithm, they use the SAR backscattering as the predictive variable for the forest biomass estimation. And but we thought that if we decompose the SAR backscattering into the different scattering powers that can generate due to the different structures in the forest, like interaction from the canopy and interaction from the woody region of the forest, and the ground and the tree stem interactions. So we decompose the total, and the another reason is that SAR backscattering only contains the intensity information of the signal, but using the scattering power decompositions, we can also use the phase information of the signal. That is very important to understand the heterogeneity of the forest. So this is the study area. This is like where we developed our model. It is the Shimoga forest in the Karnataka Western Ghat of India, and this region has the very diverse of the forest. They have the evergreen forest, dry deciduous forest, moist deciduous forest, and another forest reserve like Tundi, that is the eastern part of in the India, we have used for the cross validation of our model. So this is the general methodology of the this model. We have used the full pulsar data, L-band data, and after that basic pre-processing of the data, we have generated the 7 component decomposition models, and using this model we have generated the different scattering, like surface scattering, double bounce scattering, volume scattering, and the helix, oriented dipole, compound dipole, and the mixed dipole scatterings. And using these parameters of the scattering models, we have used the random forest algorithm with the field data to estimate the forest AGB. So first we did the sensitivity analysis, how the different scattering powers are interacting with the different parts of the forest. So here we can say that the volume scattering, helix, and the map of the mixed dipole and the double bounce scattering, they have the different scattering behavior in the different parts of the forest. And there also the correlations with the biomass is also varying. So after this, we have identified which components of the scattering power is more important to estimate the forest biomass. So then using the random forest variable importance chart also, we have identified which scattering component is more important. And after that, we have tested various combination of the scattering power to estimate the biomass, and which validated with the ground data. And it is found that the combination of the volume scattering and the most dipole scattering and the helix scattering provided the most accurate results. And the RMSE is found around 19.46%. So further to test, so our findings can provide the ability of the L-band data to estimate the forest dynamics, and it has performed the existing models based on the SAR backscattering in the tropical region. And after that, to identify the cross validation of this model, we have tested this model into the another part of, we developed our model in the southern part of the India, and then we tested our model in the eastern part of the India. So this forest is located in the eastern part of India. So we have trained our model based on the Simoga forest data, and then we have estimated the biomass in this eastern part of the India. And after that, we have validated the result with the field-based data. And then here the RMSE is found around 22.9%. So the performance of the AGB model was stable, and it can be used for the different parts of the India for the estimation of the forest biomass. So after that, we have also explored this model, how we can improve the model accuracy if we use the multiple frequency and the JDA data, because the penetration of the SAR signal depends on the frequency of the data. So C-band and the L-band, mostly they are used so far to estimate the forest biomass. So first, based on the L-band data and C-band data, we have estimated the scattering, 7 component of the scattering power, and we have processed the JDA data and interpolated the forest stand height in the study regions. And after these, we have used these 3 components and the predictable variable to estimate the forest biomass. So we have found some interest. So we have also compared the sensitivity of the different signal, what is the difference between the same power, how it's behaving with the forest. The change in the spatial variability of the volume scattering generated from the C-band of the radar set data, and the spatial variability of the volume scattering of the generated from the L-band data. Same we have done for all the components of the 7 component of decomposition power. And we found that there is a change, volume scattering dominated using the L-band data, and where the C-band data, we found that mixed dipole scattering was dominated in comparison to L-band data. So these are the results of the multi-frequency analysis, and we can see that there is a significant improvement in the accuracy of the biomass estimations. So first, if we use the single frequency model, L-band performed better than the C-band. And if we combine the L-band and C-band, the accuracy is improved significantly from 19.46 to 14.77%. And if we added JDA to further improve the accuracy, the accuracy is only marginally improved, only 14.2%. And this is also interesting because existing SAR-based models, they use the backscattering for the multi-frequency analysis, and if they combine the JDA data, there was huge improvement in the accuracy. But if we use the same with the model based on the scattering power, there is only marginally effect of the JDA data. And because the JDA data, there is a 600-meter gap in every footprint of the JDA data, so there is huge uncertainty in the interpolation. So If we only based on the SAR data, we don't include the JDA data, we can still get the significant accuracy of the forest biomass estimation in the tropical forest. So this is our future work, because we know that NISAR will be in near future, it will be launched, and it will be the dual pole data. And we also developed the scattering decomposition for the dual pole data. Most of the existing models, they need full pulsar data for the scattering power generations, but the temporal resolution of the full pulsar data is very poor, and if we want to assess the forest biomass frequently, we need more frequent data. So that's why we developed, and this work is also published, the development of the scattering power decomposition. And in the past, most of the studies, they used L, C, and X band for estimation of the forest biomass in India or other regions in the tropical forest. So S band data is still less explored. Like what will be the behavior of the S band data for the forest biomass estimation? So upcoming NISAR needs some models which can use the S and L band combination to estimate the forest biomass and multimodal capability. So here we can also see that between L and C, there is huge polometric variability in comparison to between L and X. So between L and C, it is less explored so far. So in the future, we will develop the model in the combination of the L and S band to estimate the forest biomass. Here also you can see that how the polometric behavior is, that there is a difference between the L, C and X. And we can see that between L and C, we can see the significant variation between the scattering behaviors. And here also, and there is a less difference between the L and C and X. This is the S-band data for the, and we have generated the G4U based scattering model using this S-band data, full pulsar data, and we can see that how it's variation within the forest. So this is our early results for this biomass estimation, and we have used this study area because the availability of the S-band data. And here we have used the S-band NOVASAR data, JDA data, and the field data, and we can see that there is a accuracy is around 21 point. And here we have not used the scattering power; it's only based on the back scattering coefficient. And if we use the scattering power, it can further improve the accuracy of the model. So in the future, conclusion, the scattering based biomass estimation model performed better than back scattering based, and the multi-sensor model further improved the accuracy. And in the future, we want to develop the operational product for the AGB estimation using the NISAR L and S band data. Thank you.
Okay. Okay. Thank you very much. So if you have questions.
Thank you for your talk. It's neat to see the SAR data being used in this way, but also as kind of fusing different types of SAR data also with JEDI data. We haven't been using SAR data, but we've been using JEDI data to estimate things like canopy height in other tropical forests, mostly Central Africa. And we see that the really tall canopy forests are underestimated, which makes sense, and I'd be happy to talk about that. But it was interesting because some of your earlier figures seem to overestimate biomass for low biomass to mid biomass areas and then underestimate in high biomass areas. And then that was kind of improved with additional fusion of different SAR data sets, but then kind of came back when you included JEDI data again. Can you just speak a little bit to a couple things? Why you think it's valuable to integrate the JEDI with the SAR in this case, when there could be more, we may be getting more accurate information from the SAR data than from the spaceborne LiDAR. I'm curious what you think about that, if you think that's true. And then your ground observations in terms of representativeness of different levels of biomass, and if you think it's valuable to over or under sample in different areas to improve more accurate estimation across the full range of biomass.
So the answer for the first question, we have Hello. Yeah. Answer for the first question, like we as I mentioned, there is a huge uncertainty of the JDA data, and we explored that there is a less effect on the biomass estimation with the combination of JDA with the multi-frequency SAR. Only the marginal improvement in the accuracy. So our future model, we are only going to include SAR data based on the S band and L band, and we are going to generate them like operational product from the NISAR mission. So, like JDA have that huge uncertainty of the overestimation and the underestimation, and there is a like spatial resolution is also very poor. And the answer to the second question, like we have conducted the field survey in like, oh, I have not shown the field plots, but we have, for the field plots we have converted a different part of the forest. It's not only located in only the one region of the forest, and the biomass in those plots are varying from the 20 megagram per hectare to 300 megagram per. So there is a huge variability, and also to check the transferability of the model, we have also checked in the different forest region. The model is we have developed the model using the Simoga forest data, and we have implemented that one on another, like from the southern part of the country to the eastern part of the country, and still it provides the reasonable accuracy.
Well, it's convenient you picked up on this slide. So the claim at the bottom is the model was stable and suitable for transferability. And so I see the one metric you used was root mean squared error, expressed two ways. So I guess the question is, why did you use root mean squared error? And what's your definition of suitable?
Like I use the root mean square error because in most of the existing literature for the biomass estimates.
I can't hear you.
Can you hear me? Yeah. So in the existing literature, they use RMSE. So to compare with the other models, I have used this root mean square error so that I can compare with the existing backscattering models or other models based on the optical or other remote sensing data. Suitable in the sense?
Why do you say? Did you say that that's?
Because in that where we have developed the here, you can see the root mean square for the where we have developed the model, it was the 19.46. So we have to use the same metrics to compare with the where we have implemented the forest biomass, and here only there is a only decrease around 3%. So that's why we are saying that it was stable in the both region. The root mean square error was not significantly increased after the on the transferability. And that's why error with the field data.
Okay, thank you very much. Thank you, Rajat. Yes. So let's welcome the next speaker, Professor Ram Avtar, and he is going to give a flash talk. So yes, go ahead. Go ahead.
Yeah, title of my talk is Development of Geospatial Risk Index Tool to Monitor Human Nature Conflict in Africa. So this is a recent project we got from the Japan Agency for Science and Technology. And we have partners from Kenya, Botswana, and South Africa. So we are working towards to develop this geospatial risk index tool. So I will just give you very brief information about this recent project that we have just started working on. So this is briefly about myself. I'm currently working at Hokkaido University. So before starting my talk, like why we need to work towards this human-wildlife conflict, as we know this, when the conflict happens, then it has the negative impacts not only to the human beings, but also to the wildlife. So if we look into the definition of the human-wildlife conflicts, it refers to the conflicts between human and wildlife It occurs when wild animals pose a threat or damage to human safety, property, and resources, while human also pose a threat to the survival and habitats of animals. So it means it will have both negative impacts to the wildlife as well as to the human being. And there are multiple causes of this. It's the human wildlife conflict happens because of the habitat destructions, because of the limited resource availability, and sometimes there is also change in because of the climate change, the migratory pattern of the animals changes, and also there is also the increase in the population of human beings, as well as so there is more encroachment, they want to grow more agriculture, as well as the increase in population of the wildlife. So therefore, these kind of conflicts happens, and these conflicts are quite common in Africa, and there was less attention has been paid by different research institutes. So we are looking into how we can mitigate these conflicts by developing some kind of tools by using the geospatial data. So, and this human-wildlife conflict is globally widespread, affects wildlife and humans simultaneously. And study shows that climate change has an impact on human animal behavior and migratory pattern, leading to human-wildlife conflicts. So what happens, like in case of we have been studying of the Okavango Delta, there we have noticed, like with the change in the rainfall pattern, with the change in the land surface temperature, the migratory pattern changes by these wild animals, and they rate through these agriculture crops. So then there have been like lots of the cases of the human-wildlife conflicts. And when we talk about the human-wildlife conflicts, it also leads to different kind of problems like this illegal wildlife trade, climate change and its impacts, the zoonotic diseases are the umbrella target of this study. So we are not only just looking into the human-wildlife conflict, but also other components that is associated with this problem we are also looking into. So there are these two main objectives of this project. So first one is we are looking into GSRIT policy framework, where we are trying to understand and identifying and mapping the drivers, ecological, socioeconomic, and spatiotemporal driver. Then we are also working the quantitative and qualitative study on the human-wildlife conflicts and illegal wildlife trade, and understanding the impact of national and global policies on human-wildlife conflict. And as I mentioned, we are developing this tool working with the different wildlife agencies in Kenya, Botswana, and South Africa. So we are working closely. We are looking into what are the drivers and how we can integrate all those drivers and how we can estimate the risk by looking into the hazard and vulnerability in those areas, by looking into the past incidences. So based on that, we are trying to calculate the risk by looking into the hazard and vulnerability. And finally, we will make this web-based interactive mapping that can showcase us, that can help us to predict us, okay, these are the severe sites or these are the hottest spots where it's going to be some future human-wildlife conflicts based on our past incidences. So that kind of geospatial tool we are going to work. So this is the theoretical framework. So these are the main drivers that we are looking into: the climate change, the growth in the population, the economic impacts, and what are the impacts of these different drivers, how they are interlinked, and what are their connections. And so we are looking into all these components. So we are bringing all this climatic data, the socioeconomic data, as well as the data that is coming from the land use, land cover. And we are integrating all this data in a framework to develop this GSRIT tool. And finally, we look into the risk area based on our past incidences. So what are the vulnerable areas if there is a severe change in the land use, land cover, if there is increase in the cropping area, then how it is impacting, and if there was some past incidences of the human-wildlife conflict, what was the causes of those incidences. So we are looking into all those causes. We are also discussing with the local people, local community people who are living near that areas to look into what are the drivers. And finally, we are integrating all those drivers in a GIS-based platform to develop this GSRIT tool. So this is the overall theoretical framework that we have been working on. And So and there, these are the input variables that we are going to get. So these are our 3 sites, in Kenya, Botswana, and South Africa. And we are working closely with the wildlife agencies there and officers there. And we are integrating all this data that is existing data that is coming from the national database. And also we are trying to get this real-time data set that is coming from the ground. And we are integrating all this data to develop this tool. So this is the overall framework of this study. And we have recently started working on this Okavango Delta in Botswana, where we are looking into how the land use land cover change is happening, and we have developed this remote sensing based ecological index, and we are comparing this RSEI as well as the land use land cover change with the incidences that happened in both in case of Okavango Delta. So here I just want to show you some briefly, because I have very limited time. So here we first developed the land use land cover. So you can clearly see there is a huge expansion of the agriculture farms because of the increase in the population in this area. So there is expansion of, and these are some of the migratory tracks, because from this area these wild animals or elephants, they migrate to another national park when there is a lack of water. And sometimes some of the farms, they are in between the migratory routes of these animals. So we are looking into how there are some studies that shows what is by using the GPS collar, they have tracked what is the movement of those wild animals, especially the elephant. So we are looking into whether these incidences happen when there is so these human wildlife complex locations overlapping with the farmland and village locations. And these incidences are increasing with the time. So we have also developed this RSEI, where we are looking into what is the impacts of this, to look into how this can be helpful to mitigate the human wildlife complex, and what is the relationship with the increase in temperature. So this RSEI also shows the positively correlation with the precipitation and negative correlation with the temperature. So this is some of the preliminary results we got, and we have been working closely with the Okavango Delta institutes there. We are sharing our results based on the remote sensing based data, and they are sharing the results based on this human wildlife incidents. So these are some of the results like that shows what is the monthly average MNDVI and NDVI pattern in that area, and how it is related with the incidents that happened in the past. So we are looking into that pattern. We are exploring the pattern of these. So here you can clearly see the LST, the land surface temperature, and then MNDVI is decreasing, the LST is increasing. So it shows that there is a decrease in the water resources and increase in the land surface temperature. So somehow it is also impacting the wild animals movement and migration. So we are looking closely how this change in the vegetation pattern, change in the water bodies, and how these animals are migrating from one location to another location. So there are some research we had just conducted, like the very preliminary results we had just developed. And finally, we want to build all this, we want to bring all this remote sensing based analysis as well as qualitative and quantitative data to look into how we can develop the risk tools that can help us or support us to mitigate the human wildlife conflicts. So that is the main objectives of our study, to look into how we can develop the risk tool, the GSRIT tool.
Okay.
That's all. Thank you very much all.
Okay. Okay, thank you. Thank you. Any questions? Please raise your hand. Okay, so if not, let's move on to the next talk. Okay.
Yeah.
So the next Next one is ongoing work from a colleague, but he could not come, so he asked me to deliver a flash talk for him. It's ongoing work, so I will try to make it clear. So we're trying to see is some vegetation change in a tropical dry forest with time series analysis. So the general object, sorry for the small text. Yes. So the general objective is to evaluate the trend of vegetation cover in the tropical dry forest during the period of 2016 and 2023. I think one of the reasons the data period is because he's trying to use Sentinel data. So Sentinel data is started from the state, and by using the vegetation enhanced vegetation index, so we're trying to see how vegetation cover has changed over time. A negative trend may indicate vegetation degradation. That's what he's trying to see, or loss of forest cover, while positive trend may indicate vegetation recovery or expansion. I'm not sure this is the right presentation. Okay, but anyway, so here is a general description about tropical dry forest, and as you can see in that study area, tropical dry forest has been designated to be used of shifting cultivation and cattle grazing. So there are quite a lot of degradation going on there. The study area is located in the central western part of Mexico. It's a river basin. The name is Ayuquila. In that basin, there are about 24% is tropical dry forest. The other part is like 12% is temperate forest composed by pine and oak. And the center area, the flat area, is more designated to the agriculture activity. And on the slopes are mainly distributed the tropical dry forest, which are, yes, used for the shifting cultivation activity. So here the data, yes, mainly used from Sentinel. I'm not sure this is a slide I'm familiar with, but anyway. Basically, here is a method flow. So the Sentinel data were collected. Sentinel-2 data, optical, were collected during this period. And then first, a forest mask for tropical dry forest was applied. And then the enhanced vegetation index was extracted from that time series, and then goes through some quality filter. And then we apply this kind of trend analysis, and then we kind of classify the trend like negative, positive, and allocate some no trend value range, and then goes through the accuracy assessment using the high spatial resolution images available from Google Earth, and then we finally get the accuracy assessment by error matrix. Okay, so here is, and then for the accuracy assessment, mainly for those three categories, using a stratified random sampling to allocate a certain number of random points to go through the accuracy assessment. And then here you can see the general global accuracy is around 80%. And then also we can see that for majority like stable forest, that means during that period we have not detected any change. So there are some also negative and positive, which are related to the degradation and recovery. And you can see generally the forest loss is easier to be detected by remote sensing data than forest gain. Okay. So these are not. So here is the result of the trends detected. So the red color depicts the area with negative trend, and the green color depicted the area with positive trend. And then here there are some specific. Okay, there are some cases representing the some force change. For example, those areas detected as change, but they're actually the forest changed to agricultural fields, permanent agricultural fields, since there is agriculture activity going on, so detected as if there is a positive trend. Here is a negative trend detected, as you can see. In the beginning period was covered by forest, and the next is a forest loss, which is being used as shifting cultivation. And those are some examples as no trend area. And so the general conclusions is there are more, in general for the study area, there are more negative trend area detected than the positive trend. So there are more like seemingly indicating that more forest loss than forest gain. And but we believe that since there are some false changes going on, we need to, the next step is to focusing on those areas to really to study it a bit furthermore. I think that will be all. Thank you very much. And if you have any questions, please let me know. Yes, please. Can you give that?
Hi, I'm just curious to learn how did you create the mask that you use as a first step to isolate forest from everything else?
Yeah, thank you. So that is a mask is created by a classification of Sentinel-2 optical images, but we didn't do the classification. It was from an existing map from the state environmental agency. They provided that map. Yes, thank you.
Yeah, any other question?
Yeah, thank you.
So on your point 3, you present overall accuracy 80%. Why
Yes.
did you select that metric to highlight?
So that metric is mainly composed by those ground control points, and then for those 3 classes, like certain points located as negative and no change and positive, and then then calculated as over accuracy. I don't know if I understand.
Why select that particular metric to present?
Right. Actually, I believe the reason is those accuracy assessment, it can indicate really between forest gain and forest loss, which one would have better. Actually, what we would, the person, I mean, the colleague wanted to emphasis is to show the accuracy for the gaining trend and also the losing trend. But I don't think the overall accuracy is that important. Yes. Yes, you're right. Yes, you're right. So these metrics, yes, you're right. Because in the general overall accuracy, it might because there is a dominant class which has high accuracy. It might mask out those rare classes that has much lower accuracy in comparison. I think they might have some other metrics such as F score would probably better represent.
Give the 9 numbers of the center of that matrix, and people would compare the 90 to the 78 and 155. And then we say, oh,
Yes.
I don't need to say more numbers than that.
Yes. Yes, I think that is one of the maybe disadvantage of using this error matrix to represent, to do accuracy assessment for the rare classes. Yes.
Well, I love the matrix.
Yeah.
Need the summary
Okay. You mean the overall accuracy is not a good indicator for this? Right. Okay. Yes. Yes. We'll take care of that. Yes. Thank you. Okay. Yes. Thank you.
Yeah, thank you very much. Yes. So now let's move to our next presentation. So I would like to invite Gibran Oliveira. So the presentation is about monitoring climate change impacts on dryland biodiversity. So if you are ready.
I need us here, just checking. All right. So first of all, hi everyone, I'm Gibran, and I'm here to talking about my work team project. The title has monitoring climate change impacts on dryland biodiversity from Brazil. So you already know about the climate change projection, and we will increase the temperature. And we already know also about the mainly cause of extinction risks on species, mainly on mammals and some plant groups. And in the Caatinga, it's a dry land from Brazil. It's our biome. It has the projection estimated like a reduction of 50% in the precipitation and decreasement of 4.5 degrees in the temperature. This dry land has the size of the Mexico. It's about 1 million square kilometers, and half of this is already altered by human activities. So some species can respond to these climate changes, expand the distribution or doubling it. And other species can just shift the distribution, while another ones can be just extincting locally for their communities. And it will just change the community structure, and most of these impacts are due to the introduction of new species and the local extinction. So it can just reduce the richness and can reduce also the dissimilarity, so create homogenization. And we know that those species are very important to balance the ecosystem. And the reduction of these species can just disrupt the ecological service and cause the damages like diseases spreading, venomous species, pest crops, and even altering the water cycle. And it's already happening with exotic and alien species. So to know that, we need to map the future suitable areas. And we did it just using ecological niche models and species distribution models. And to do that, we know that ecological niche models will just show the suitable areas. So we use the species distribution model just to know the accessible, suitable areas to these species and these communities. So to do this, we use this package in MTML, and we just do a lot of processing, and we just pre-processing, processing, post-processing, and we start with input data, we remove like coloniality, we just clean the occurrence data, we also attribute some pseudo absence, we define the accessible area, we just did the extrapolation to estimate the suitability, and we did a data partition, but here we don't use bootstrap, we just use scaffold because we think it's work better and more quickly. And for the algorithms to define the suitability, we use the approach of ensemble. So we use three classes of algorithms. We have here envelope class, we have here statistical class, we have here artificial intelligence. So after that, we just define the suitability. So we have here the niche communities. And after that, you just apply the mask of restriction and to define the species distribution models. So we use the sovereignty index because we are working with the only presence data. So it's important because I guess it's just give more weight to this type of data. So we define the binary maps after it all using 5 generalized circular models from the IPCC and the pessimistic scenario and the most optimistic one. And we are defining at first for the current time and to the meaning of years of 2016, 2100. And for plants, we just got at the end of the all processing, just 300,000 records for almost 3,000 species. And most humbly, I just got 20,000 records covering 995 species, including new species that was out of the data set and out of the literature. So here is the main reason, because we use these three class of algorithms, just to use the advantages and reduce the negative impact. Mainly the interpretation has artificial intelligence, so there is some estimations that are a lot of simple, and there is estimations that have intermediate precision. So doing that, we can just have the meaning of everything at all. And to define the taxonomic diversity, so we just divide in grid cells, that is our communities of 100 square kilometers, and for each grid cell we has a value of species number, that the richness, and we have also the similarity value, that's the beta diversity, and we derive from this value for the future a delta value, so we can just make this to know if the value is negative, is attributed by the loss of species or the homogenization. This first work with plants, they were published in the Journal of Ecology, and it's very interesting because it's really pervasive, as the title said. And we can see in the top just the richness and all the alpha diversity. So we can see the current time and the future time. So here we have 100% of species loss, and not only for the optimist, but for the pessimist. And for the beta diversity or the similarity, we have 40% of homogenization of the Caatinga land and 30% in the pessimistic scenario. So you can also just highlight here that the richness and the most dissimilarity areas will be the areas that will lose this type of diversity. So in this box plot, we can just see the state of this community. So in the top of it, just the species richness, and we can see that the community with more heterogeneization will have more species, all right? You just see the red one. But when we are comparing with the bottom part of the graph, when you divided the delta value, all communities will lose species, and it's not only the homogenized one, but mainly the heterogeneized one. It's an interesting process, and I can talk with you after about that. And we also compare this with the wood species. So we can see that there is a real loss of wood species and a real increasement of wide range species or generalist species. So here is a map of this process for both scenarios in the future. So we'll just lose all the trees that we have in this biome. And the second part with mammals, we just published in Global Change Biology. And we got this data, so it's the same thing, just characterizing the communities. So we have more species in lowlands, is very interesting. And we just believe that is the main reason, because the body mass too. So they also will have more body mass species in those lowlands communities. And in general, it's very similar. But when we see the map here, in the top part of this, we can just see the richness, and we can see the similarity, and we can see the body mass in the top. So in the bottom, there is the delta values for each one of them. So we can see 80% of species loss to the pessimistic scenario in the future. And the same thing, the same pattern, if you can look, so the most richness areas and diversity areas will just be the regions will happen more loss of species. So for homogenization, about 70% of the communities of the Caatinga will suffer with the homogenization, and it's interesting result, but the Caatinga will increase the body mass, and I will let you to ask me about that with more ease to explain. But here it is. I have the pleasure not only to run the model, so I could go to the field work and collect some animals, and it was very interesting, because here we just created a list for each species. There is the 95 species here. And here we have the log of body mass. So it is the most dark one is just more body mass, and the white one is just less. So we can see a lot of loss of range areas for small species, and we can imagine it is because of the capacity of dispersion of these species. But some species are not excluded from that, like large species, and we must to think like those species that are large and have a wide range dispersion, we also need like shelter and things like that. They are the most hunting species, so it's not safe for everything at all. So including if you think about the data of the plants, so they are trophic connection between them. And so in the end, we conclude really the climate change will lead the drylands of Brazil to homogenization and to richness loss. But at last, but not at least important, we also work not only for outside to inside, but we also work for inside to outside. So we visit few communities just to talk with them. We have support for this program. It's a very interesting program. We are trying to ensure resilient communities with energy safe, food safe, and water safe in one of the driest regions in the Brazil. So those people, they just love what they do. They just love their lifestyle, and they just need quality of life. And it's not at all only about money, but how they construct their happiness and everything like that. So even living in biggest capitals in Brazil and another place, those people just want to keep their way of life and pass this for generations. And we want to avoid this type of scenario. Desert is not natural in Brazil. So it's a desert in the Caatinga, provoked just by the activity of the society. And so I had the opportunity to visit a few of those communities, to talk with smallholders and talk to them about climate change, but not only about that, but to hear them, to hear their problems and how we can help them. And this project is going on right now. And so we went into some schools too, to talk with the children. We just got together with another researchers too. It's a really, really great project. So we are like collecting a lot of biodiversity groups, and we are trying to understand how biodiversity at all in landscape will help those producers to have a high productivity and keep their lifestyle. And after that, we just went to some divulgation, but also in the universities and colleges and schools. And we also achieved some awards and honorable mentions, just to highlight the dimensional size honorable mentions, and some awards from the Climate Reality Project. And we were shortlisted in the postgraduate awards from the Fine PhD. We were the only one from the Americas, right, in that list. And I'm really pleased, and I'm really thankful to represent my partners, my friends, and my communities. And I look forward for your collaboration and for your questions. Really thank you.
Thank you very much. So any questions? Please let us know. Ah, yes. Okay.
I am really curious about how you are connecting all the science that you presented in the first half of your presentation to the more, I don't know if it's like training or activist or actor engagement part of the second part of the presentation. What are the challenges that you have faced in linking those parts? What are the, I don't know, like personal, maybe satisfaction that you get from that? If you can speak about it a little bit.
All right. The second project, named Next Caatinga, right, is not a new initiative, and it's very connected with researchers, associated professors that are from those regions. So it's very interesting. They already know about some problems, and they went there before, so I was like invited after I finished my master degree to go there and just help them. And at the end, I just get lucky to connect my master degree with them because I just did all my master degree in pandemic, so I was in period computer and it's not my way of life to do, so I was very lucky for that, and we just did that connection. And right now this program is still running in my state, so there is a lot of research too. So what is very interesting is every person here just a PhD, a master degree student, so they are just looking for the project, and we have this close contact with the community. And what I felt is like we try to sometimes teach the people, drill them, and just take our knowledge and trying to throw them and do whatever we want with that. But what happens here is that we just visit all these communities and collect data from in some sites. And when a neighbor is seeing his neighbor working and everything going good in their property, in their production, so they start to be curious, what is happening right there? I want you to research my land too. I want to make the difference too. So what I learned for that is that they are changing by example. We cannot teach in just talking. We need to show to them the example. So I feel those communities are changing, but they still in process. All right. So when I get that with those people from those projects, they just told me that we needed like to listen because this type of thing is very common to them. But there is another alternative they used to do, like changing the cropping and turning it with another type of cropping, avoiding to use the fire. But most of people that listen for this type of behavior or production, they used to say you are lazy because you let the land resting, you are not using the land to produce, just to forest. And the rainy seasons and everything around is just changing. So I do believe that little by little, this community is changing. My main goal has not only has a research, but has a person is just to create also resilient communities, and not only in the paper, but also in my community. That's it.
Yeah, thank you. Thank you very much. So any question? Some other? Okay, so let's move on to our next speaker.
So thanks for the opportunity to present today. I'm Hannah Stouter, and I'm a PhD student at UCLA, and I'm going to be presenting on work we've been doing to look at patterns and drivers of forest and land cover change in Central Africa across a few different scales. And so the first thing I want to do is this project has really been a huge team effort, and I just want to shout out the whole team, both collaborators in California and in Cameroon that we've been working with to really make this project happen. So global change is impacting all over the tropics, but research is increasingly finding that the Congo Basin is responding differently to global change than other tropical forests. And so when we just look at trends in the in the strength of the carbon sink from about 1990 to present, we can see that while we've seen this decline in the Amazonian carbon sink, we're seeing relative stability in the African carbon sink. And when we look at forest plot data, that's also looking at the impact of the 2015-2016 El Nino on above ground biomass, what we see is that despite changes in increased temperature and decreased precipitation, we're not actually seeing changes in the above ground biomass during this kind of climactic anomaly, which caused the authors of this paper to conclude that there might be some kind of resistance to African tropical forests to global change. This similar work was done to look at the impact of the 2015-2016 El Nino using spaceborne remote sensing data, and this research did find that across the tropics there was a temporary carbon flux, so all those carbon stocks reversed, but that the drivers of that change varied across each continent, each tropical basin. And so we're seeing that the impacts of these global change are actually differing in the tropics, and in the Congo Basin in particular, maybe responding differently. And one reason research has pointed to that could result in the Congo Basin responding differently is the main driver of land use in this region is small-scale agriculture. And this really is different than elsewhere in the tropics, where we're seeing much larger deforestation processes for commodity crops. In the Congo Basin, we're seeing small-scale agriculture related for subsistence and agricultural purposes, largely, or rural, local kind of development on a much smaller scale. And this varies a little bit across the basin, but this is still the primary driver that's driving land use change in this region. And so with this in mind, this project really wanted to kind of focus in on exactly how land use is changing and how forest is changing, and we wanted to do this at 2 different scales. So at a landscape scale, we're working in this 300-kilometer region in Central Africa centered on one forest reserve, but this is a region that encompasses a diverse mix of different land use types. So we have large centers like Yaoundé, which is the capital of Cameroon, but it also includes this Tridom region, which has several important conservation sites. And so at this landscape scale, we're interested in comprehensive forest mapping, so creating high confidence maps of forest disturbance and then also forest gain, as well as mapping patterns of land use change from 2000 to 2024. And then we want to combine this work with trying to understand the direct and indirect drivers that are shaping land use change at a local scale. So this is work that's taking place right around the Jah Reserve, which is that reserve at the center of the 300-kilometer area. And here we've conducted household surveys to try to get at what the direct and indirect drivers of land use change might be, and then start to untangle the role that conservation and livelihood projects might play in shaping land use change at a small scale. And so as I said, this is part of a much larger project, and there's a lot of different components, and I'm really going to be focused just on these 3 parts of the project. So in terms of just a quick overview of the methods, for this comprehensive forest cover mapping, what we've done is we've taken these global data sets, global or pan-tropical data sets that look at either forest loss and forest gain, and we're combining them and looking at pixels that show overlap between those to then get a better sense of where forest loss or forest disturbance or forest gain may be occurring, and then conducting accuracy assessments both on each of these individual data sets, but then also on a combined data set. For the land use land cover change mapping, we've developed a regionally specific labeling data set across about 6 different land use types, and then we're building out a random forest model to actually predict land use change across the entire 300-kilometer region. For household surveys, we conducted over 300 household surveys, and we mapped over 700 fields around the Jah Reserve, and these surveys included questions about agricultural practices, about project engagement, and then about perceptions of ecosystem services to start to get at those indirect and direct drivers of land use change. So moving on to some of the results. So in terms of forest disturbance, what we're seeing when we look across all 3 of these data sets is we're seeing slightly different patterns of forest disturbance across the region. We are finding that when we combine these data sets, we're getting a more accurate understanding of where forest disturbance is actually occurring. But when we look at this trend over time, we're seeing that the patch size of forest disturbance is increasing, which is interesting given that we know this is a system that is really driven by small-scale land use change. In terms of forest gain, we're seeing a similar pattern where different data sets spatially are showing gain in different areas. And we're also seeing that these data sets may be picking up gain differently in different land use allocations. And so especially thinking about forest gain, different data sets could be using different methodologies, which could be pointing to different types of forest gain across this region. And by combining them, we might be able to get a better picture of where that forest gain is taking place. But this work is still in its early stages. For the random forest model that we've developed to map land use change, we've developed labeling for 6 land use types, so forest, mixed agriculture, plantation agriculture, built environment, bare soil, and water. And so far we've just run this model over a much smaller area at the center of this 300-kilometer region over Yaoundé, which is the capital, so it's a region we expect to see several different land use types. And so far we've just included Landsat, so all bands from Landsat and EVI, but we will hopefully include the additional data sets in red into the final model. And then we predicted this out over different numbers of land use categories, ranging from just forest and non-forest all the way to include all 6. And so what we see when we do that is a more rigorous accuracy assessment needs to be done to really expand this out and understand where those errors might be. But overall, we're getting an accuracy of about 0.87. And when we looked at the users and producers accuracy, the most common misclassification we were seeing was between oil palm and forest. And there are currently no textural metrics included in the data, and so including that may help get at it, as well as really going through the labels we currently have and cleaning them to make sure we're more accurately able to detect differences between forest and oil palm. Moving on to the work we're doing at the small scale. So this map here shows all the villages we conducted surveys in. So we conducted surveys in over 30 villages. And the size of the circles corresponds to the population and the color to ethnicity. So all the blues are Bantu ethnicities, and yellow are Baka ethnicities, and ethnicity is complex in this region. But we can think of Baka communities as indigenous communities. And what we're finding from talking to farmers is that land use change is still really driven by small-scale agriculture, but it's not only to meet subsistence demands. There are some market pressures, but they're hyper-local, and it's just people selling within or kind of between different communities. And we are seeing kind of increases in cocoa farming, but it's pretty small scale and shade grown, so it's likely going to be difficult to map in this region. And then looking at those conservation and livelihood projects, so here the size of the circle corresponds to the number of projects in this region. And this is a region that has had a lot of projects take place over the past 40 years. But no real assessment of what the impacts these projects might be having on land use change. And the majority of these projects really have to do with wildlife conservation or preventing of bushmeat hunting. But because bushmeat hunting is such an important protein source, they often have these indirect consequences on land use by encouraging people towards different kinds of agriculture. And so that's what we're trying to untangle here by comparing villages that have less NGOs with those, or less projects with those that have more, to try to get a sense of what the differences in land use change might be and how and what role these projects might play in that in this region. So some of the key takeaways we're finding from this work are that by combining different data sets, especially in this region, we're starting to get a better understanding of where forest change is actually occurring, both in terms of forest disturbance and forest gain. And we're seeing that despite the fact that this is a region that is still really governed by small-scale land use change, the size of that disturbance is increasing. We're also finding that the majority of agricultural substantiation is for either subsistence or livelihood purposes. About 2/3 of what people produce they're consuming for their household, and about 1/3 they're selling. We're seeing that cocoa could play an increasingly important role, but it's still small scale and it's difficult to map because it's shade grown in this region. A lot of farmers also brought up the impacts of changes in the start and end of the rainy season impacting when they can plant and their ability to harvest those crops. So linkages between land use change and climate change could be increasingly important in this region. And then finally, conservation and livelihood projects may have these unintentional impacts on land use change, and that may vary regionally based on the number of projects that exist in those regions and the other factors that could be influencing land use change. In terms of next steps for the forest gain mapping, we're going to finish the accuracy assessment and hopefully build that out to examine where some bright spots of forest gain and forest restoration might be occurring in this region. For the land cover mapping, we want to extend the spatial and temporal scale of that map to cover the entire 300-kilometer boundary, and then include some additional data sets to hopefully improve the overall accuracy of this model. And then in terms of the household surveys, we want to link those local drivers with regional patterns of land use change by taking the delineated field information we have and combining it with the larger scale maps we're going to be making. And then we want to examine the role that conservation and livelihood projects play in influencing these kind of patterns of change. So thank you, and thank you to everyone who's helped with this work. I'm happy to take questions.
Thank you very much. So, yeah, over there.
Hello. Hi. Thank you for your presentation. That was awesome and very clear. I just had one clarification question. So when you talk about the different villages that have different projects that are ongoing, and trying to see whether that affects perceptions or whether that affects land use change, I believe is what you were trying to determine. Did you find those projects through the household surveys, or did you do some kind of like analysis beforehand? Because that's something that we run into as well, is just like what is already going on the ground that people have already been involved with and how that affects their perceptions of.
Yeah, it's a great question, and I'd be happy to chat about it more too. So we did include questions about different projects in the household surveys, but we separately, so we would talk to individual households, we also talked to chiefs in villages, and I had done some work prior to conducting the surveys where I talked to individual NGOs that we knew were working in the region. But it's tough because villages wouldn't always tell us what was going on. Not every household's involved in every project. So the results we have probably aren't a comprehensive understanding of all the projects that are currently ongoing, much less what's happened in the past. But hopefully we can get some sense across all the villages and just regionally that there may be some of these differences. But I'd be happy to talk about that more. And yeah.
Thank you. So some other questions? Okay, very good. Thank you very much. So let's move on to the next speaker. Thank you. Who is that? Mine? Okay. So the next one is my talk. So it's also a flash talk. So I'll keep it concise and brief. So. This work is about estimating forest degradation by capturing biomass change. So we get some insights from a tropical dry forest. Here is some contents. In this presentation, we'll talk about tropical dry forest, why we focus on that, and also deforestation, forest degradation in tropical dry forest, and how we upscaling biomass from the field measurement using a combination of sentinel images and. Yeah. So why tropical dry forest? So it is one of the 14 main terrestrial ecosystem. So it also provides a lot of important ecosystem services. Here is a map about global distribution of tropical dry forest. We can see that in Mexico, it is mainly distributed on the western edge, western border side of Mexico. And here is also a brief definition of tropical dry forest. It is forests occurring in tropical region, characterized by pronounced seasonality in rainfall distribution, resulting in several months of drought. Here is a map of distribution of tropical dry forest in Mexico. I want to call your attention here. Sorry. There's some legend here. Sorry, it's in Spanish. But from this legend, you can see that the green color depicts the primary status of a tropical dry forest, and the rest of the color depicts the tropical dry forest already being disturbed, and in the secondary forest type. As we can see that. Through this zooming in window, we can see that most of the tropical dry forest in Mexico is actually in a very degraded state. So the motivation and objective of this work, the motivation is tropical dry forest is constantly threatened by deforestation and forest degradation, and above ground biomass is a direct indicator of forest degradation. However, so multi-temporal biomass data is often not available, which hinders forest degradation estimation. So in this work, we try to employ this. We try to use multi-temporal field forest measurements data to gain insights into forest degradation estimation. So here is our study area. Those I want to call your attention in those the distribution of field sampling plots. The red dots representing the 43 sampling plots in 2019, and the blue dots represent the distribution of sampling plots in 2022. So the method we use briefly is we first used multi-temporal biomass data estimation from field measurement, and then we upscaling this biomass we mapped the biomass using Sentinel data, Sentinel-1 and Sentinel-2, and also a random forest regression. And then we obtained this biomass change map, and then through that, we estimated forest degradation and some regrowth. So here is forest attributes data. Forest attributes data we obtained from the field measurements. And on the right side, you can see the lower part of the figure is our sampling map. I mean sampling. So for each plot, it covers around 500 square meters, and we divided each plot in the inner circle and the outer circle. So inner circle with a radius of 3 meters, we measured all trees with a DBH more than 2.5 centimeters. For the outer circle, we only focused on the trees that more than 5 centimeters. We measured those trees. And then so the forest attributes we measured, including the DBH and height and canopy cover. We also counted the number of trees and also stems in the plot. And then we applied this allometric equation to obtain the biomass data. So here is a distribution of the field measurements comparing this between 2019 and 2022. We can see that the above ground biomass and basal area, actually in 2022, it appears that there are a little bit higher the above ground biomass and basal area than 2019, but the rest of the attributes, for example, canopy cover and also the stem density and the tree density are actually, canopy cover has decreased, but stem density and tree density increased. We believe this is a sign of forest regeneration. Young trees are coming. About biomass mapping, we derived those variables from the Sentinel data, including the spectral and texture variables, and also spectral bands and DVI, and also texture data for those 2 dates. Here is the biomass map we obtained in 2019 and 2022, and from that we derived the biomass change, the map of biomass change. And here is a general statistics of the biomass change map we obtained. So we can see that then we relate those negative change to forest loss / degradation, and positive change to positive change, especially more than 7.6 ton per hectare to forest gain, and then a range of biomass as no change. So for accuracy assessment, we used a stratified random sampling. We locate certain number of points to those classes, and then we get for forest loss 77% of accuracy, and forest gain relatively poor, much poor, 34%. And then conclusion so far is still ongoing. So forest loss had higher accuracy than forest gain, in a way like indicating different capabilities of remote sensing in estimating forest degradation and regrowth. And also, although there are forest recovery, about 1.3% of the tropical dry forest in those areas. So overall, the forest area presented a negative trend, about 4.3% of the area. So that is all. Thank you very much.
Yeah, any question or comments? Yeah, please.
Hi, good afternoon. Thank you so much for the presentation. I was wondering, did you measure the same field plots both in 2019 and 2022?
Thank you. So that's a very good question. No, we didn't. we didn't. We try. We know the importance of observing the same plots, but it is often difficult to locate because of the same plots for the next measurement. Besides, the plot area are often disturbed for the second revisit. So we used a space for how you call that? We extended. We measured a different area, space for time. We used this kind of strategy. They have certain disadvantages, but in our case, yes, we hope it works. Yes, thank you.
So, but then the difference that you are showing there might be between sites differences rather than over time changes, aren't they?
Yes. We used 2 times of field measurements. We used as a training data. We trained 2 different models, and then the idea is to get a biomass map from those 2 dates, and then we can obtain the biomass change. So yeah, that's thank you.
Okay. So you have a continuous variable, but then for your accuracy assessment, you did you changed it to a categorical variable. Why?
Yes, that is to facilitate the accuracy assessment process, because we want to relate the positive change and negative change to degradation and regrowth. So we need to classify them, and then we can compare. Yeah.
But why do you need to classify them?
Because
if they're negative, it's loss, and if it's positive, it's gain.
Yeah.
And it's a continuous variable.
Yeah. So the idea for the accuracy assessment is to locate certain points to the category of degradation, or to the category of biomass gain, and also to the category of biomass loss. So in this way, in the categorical map, it helps us. I mean, it's easier to allocate those stratified random points for the accuracy assessment.
Okay. Well, you could talk to the first presenter who had a scatter plot. So you maintain the information that's in the continuous variable.
Okay. Yes. Thank you. Thank you.
Yeah, thank you, Jan. So now let's move to the next speaker. So next speaker is Teiji. He will present about sustainable ecosystem services, human disturbance, carbon storage, and recruitment in Malagasy tapia woodland. So Teiji, floor is yours.
Good afternoon, everyone. So it's a pleasure for me to be here and present you this work entitled Sustainable Ecosystem Services, Vegetation Structure, Carbon Storage, Human Disturbance, and Recruitment in the Malagasy tapia woodland. This is the fruit of a collaborative effort between the Department of Biology and Ecology of the University of Antananarivo, the Socio-Ecological Observatory for Studying Southern African Woodlands, and the Swedish University of Agricultural Sciences. Madagascar is a country that has a lot of biomes, and dominated mostly by the savanna. And in the savanna biomes, there is a particular woodland named tapia, and its occurrence zone is from the central to the south-central part of the highland. And as ecosystem services, this ecosystem provides timber and firewood, medicine and fruit, and it is also the habitat of a white silkworm called Porosera kajani. This ecosystem is threatened mainly by the recruitment difficulties of the main species, which is Wapakabojeri, colloquially named tapia, the over-harvesting, and exotic tree species invasions. And then from these threats arise the following problems. Savanna woodlands store a certain amount of carbon, but its relationship to vegetation structure remains poorly understood. And Wapaka bujeri species have a poor recruitment by sexual reproduction. To fill these gaps, we address the following research objectives. First, we describe the vegetation structure attributes of Wapaka bujeri woodlands by installing long-term vegetation plots. Secondly, we studied the relationship between vegetation structure and carbon storage by measuring above-ground biomass. And third, we investigated the recruitment issues of Wapaka bujeri, whether it is due to a low seed availability for germination. Our study site is located in Arrivemamo. It is in the central port of Madagascar, within the province of Watanabe, in the Arrivemamo region, belonging to the Arrivemamo municipality. We selected 5 sites where installed the first 5 1-hectare suso plots for Madagascar. These plots were installed along a human disturbance, which is the distance from the main road. To measure the vegetation, we measured trees by measuring height and diameter at breast height. We also counted the number of shrubs within 3 2-meter transect belts, and along this transect, we also counted the number of fruit in each fruit and trees. And for herbaceous biomass and canopy cover, we sampled the herbaceous layer in 0.25 meters square quadrats, and within those quadrats, we counted the number of fruit on the ground. And for canopy cover, it has been sampled in 2 perpendicular transects. We performed the following data analysis from this data. First, we used descriptive statistics of vegetation attributes. Then, for the second one, we tested the relationship between vegetation attributes, carbon storage, and distance to the road. And third, we modeled the factors that could be influencing Wapaka bujeri's recruitment. Among our vegetation attributes, canopy cover, stem ratio, and the mean herbaceous cover showed a positive trend to distance. Diversity showed a negative trend as it decreased further away from distance. And the previous variables that were mentioned were also correlated to a positive trend to carbon storage. And about the recruitment, further from road, we could observe more fruit on the ground. But the amount of fruit on the ground did not influence the number of seedlings in our quadrats. Then these points raised several points of discussions. As we observed low tree species diversity, a variable vegetation coverage, stem ratio, and herbaceous cover. In fact, human disturbance then influences the vegetation structure by agricultural expansion, frequent fires, and selective logging. There is a positive correlation between vegetation structure and distance to the road in the Wapaka bujeri woodlands. Less disturbed areas allow the vegetation to develop, and structural attributes like canopy cover, stem ratio are related to higher carbon stocks. In other various ecosystems, structural diversity is recognized as a determinant of carbon storage. However, more data are required to draw strong conclusions from our trends. And the species Wapaka bujeri have a poor recruitment. This is mainly due to its poor germination. This is partially attributable to the absence of its disperser that could have gone extinct due to human anthropization. Then, if we want to support the continuity of ecosystem services within those woodlands, there is the need to minimize human disturbance by protecting the original landscape. but also more geographic coverage and a long-term monitoring is needed, for example, integrating satellite data, and improving the colonization of Wapakaborgiri species is necessary. Even the past main primary disperser are gone. Species such as birds and bats are good dispersers, but they are often overlooked compared to the other species like lemurs and mammals. So that's all for my presentation. Thanks for your attention.
Yeah. Thank you, Tenri. So now floor is open for question and answer session. So is there any comments or questions? Yeah, please.
Yeah, thank you for your presentation. It's quite interesting. Can you quickly show where you are taking, someone is taking measurement DBH? So I saw this picture and it's so interesting. DBH actually means diameter at breast height. So if this is how you took your measurement, don't you think it may affect your calculations?
Yeah, please.
I think,
yeah.
So I think most of the studies on biomass choose the point of measurement as 1.3 meters. So that is considered as a standard measure of point to measure DBH. But you know, Malagasy people, some are short, like me, I'm not that tall, so that's why it is higher, but it's at 1.3 meters height from the ground. So does that answer?
Yeah.
Yeah.
Any other question? Quick comment? Yeah.
Hi, Tendri. I am just curious, who owns the forest that you are studying?
So currently, in this part of Madagascar, this is not yet in all legal protection, but in some areas, like in the just a minute. In the other areas, in the two southern parts, in Ichim and Isal, they are already under protection. They are within protected areas. But in our study site, it isn't yet, it didn't get legal status of a protected area yet. So people keep on people keep on using the forest and converting it to pastures. It is currently only in a temporary protection, but it is not well protected yet. So it's difficult to answer in a relevant way who owns the forest.
Yeah, thank you. Thank you very much. So now let's move to our last presenter of this session. So the next speaker is Ayana, and her talk is about exploring the links between landscape complexity and ecosystem functions in forested ecosystems. So floor is yours. You can start your presentation.
Hi everyone, my name is Ayana St. Rose, and I am from the Nature Isle of the Caribbean, Dominica. I am currently a postdoc at Oregon State University, but today I will just be presenting some of my work from my dissertation. So we'll be talking about integrating structural and biological complexity into ecosystem models to improve prediction of ecosystem functions. And when I say biological complexity, I'm talking about biodiversity, so Shannon diversity and richness. And for ecosystem functions, we'll be looking at ecosystem respiration and gross primary productivity. So this research happens in forests, not just because I love trees, being from the nature aisle, but because forests are important ecosystems. They provide us with many different ecosystem services, from supporting services to cultural services and regulating services like carbon sequestration. But with climate change, we're seeing an increase in the amount of storms and the intensity in storms and different natural disasters like forest fires. And this is affecting the structure and the function of these ecosystems and ultimately affecting their ability to cycle carbon. So studying these relationships are very important. To look at these interrelationships, we're using data from the National Ecological Observatory Network, which is powered by the NSF, and it is a continental scale ecological observatory network that collects data from observational data to remote sensing data and even instrumental data. And here we can see some of my sites on your left, my right, and just how the sites are set up. So all the sites should have a setup like this, so it makes comparing and different analyses easier. So scientists have been looking at plant diversity as an obvious point of in ecosystem function research, because plants are the obvious source and sink of carbon dioxide. And more recently, they have been looking at how canopy structural complexity affects ecosystems' functions. And we can say this is partly due to the availability of remote sensing and new techniques like LiDAR. And for those who are not as familiar with LiDAR, here we have a plane that emits a sensor and it returns back to the sensor in the plane and yields products like we see on the right. And we can condense these points into digital elevation models, surface models, and canopy height models, which take up less space and less computing power when we're calculating structural complexity metrics. For this research, we calculated 9 canopy structural complexity metrics, but to not bore you, I'll just explain the 2 most important. So we're going to have like a fake forest, and first one is maximum canopy height. So this is the maximum canopy height in our site, and for this study we used the 95th percentile height to limit outliers. And then we also have top rugosity or canopy rugosity, and this is the standard deviation of canopy heights, so it's just a roughness measure. We also calculated some terrain complexity metrics. So again, maximum elevation, and then we have terrain rugosity, which is the variation in the elevation points within our sites. So we also created a combined rugosity metric that explains the variation in both terrain and in the canopy, because terrain has the ability with canopy to affect the microclimates within the sites, and this can affect biodiversity, ecosystem function, and so on. So here, just to make it easier to understand, we have gross primary productivity on our y-axis, and we have terrain rugosity in plot A and canopy rugosity in plot B on our y-axis. And we can see that gross primary productivity decreases as terrain rugosity increases, and increases while canopy rugosity increases. So we scaled these metrics, and we took the reciprocal of the terrain rugosity, and we multiplied that by the scaled canopy rugosity to get our combined rugosity, which explained more variation than its constituent parts. So scientists like Go et al. Have shown that productivity increases with top rugosity and also with plant Shannon diversity. But we have seen less studies about considering terrain or different species. So to address this gap in the literature, we are asking two questions. One, do ecosystem functions change in response to change in complexity? And two, can the observed relationships between complexity and function be leveraged to develop predictive models for ecosystem functions? So this is just a workflow of our methods. We source the data from NEON, so we use beetle, ground beetle data, bird, and plant data, and we calculated the Shannon diversity and richness. We also used their elevation LiDAR data to calculate the structural complexity metrics, and we sourced environmental and geographic data from them. We also used eddy covariance data, which is co-located with all the NEON sites, and we aggregated the half-hourly ecosystem respiration and gross primary productivity to an annual value, which was, and these ecosystem respirations and GPP were partitioned from nighttime respiration from NEE. And then we ran our models and different analyses. So here we have this Spearman correlation of ecosystem respiration on our y-axis and then our predictor variables on our x-axis. And I just want to highlight that structural, biological complexity and canopy structural complexity were all positively correlated with ecosystem respiration, while terrain rugosity was negatively correlated with ecosystem respiration. Also, I want to highlight that even though plant Shannon diversity and plant richness dominates literature, it was not as important as multi-species diversity and also beetle Shannon diversity. The most important variables here would be mean annual precipitation, mean annual temperature, beetle Shannon diversity, and a combined rugosity metric. So just to take a better look at this, we'll look at the bivariate plots. And here we could see that we have ecosystem respiration on the y-axis and our variables on our x-axis. And we could see that beetle Shannon diversity, combined rugosity, mean annual temperature all have a sort of linear increase with ecosystem respiration, and mean annual precipitation has the strongest relationship with ecosystem respiration. But I'm going to focus a little bit on mean annual temperature, because usually ecosystem respiration is modeled as an exponential function of, ecosystem respiration is modeled as an exponential function of temperature. But we see that even when we used the best model, which was our polynomial regression model, mean annual temperature did not do the best job at predicting ecosystem respiration. And this is seen in literature because it's very difficult to model ecosystem respiration. So when we added mean annual precipitation and beetle Shannon diversity, we saw that the predictive capability increased. So in the random forest, there was a little bit of over-prediction on the lower end and under-prediction on the upper end, which was surprising because a lot of literature used random forests when it comes to modeling ecosystem respiration. And the polynomial regression model did the best. Moving on to gross primary productivity, we see similar results. And surprisingly, again, beetle-Shannon diversity comes up as a very important variable, and we have mean annual precipitation and a combined rugosity metric. Looking at the bivariate plots, very similar, with mean annual precipitation as one of the top predictors. Oops. And here we see that again the polynomial regression model is doing a better job at predicting gross primary productivity, and these predictions are a little bit better than the ecosystem respiration predictions. So in conclusion, we can say that beetle-Shannon diversity and combined rugosity are strongly correlated with ecosystem respiration and gross primary productivity. Now this came kind of a shock to us to see beetle-Shannon diversity appearing in this equation, and we still have to do a lot of research because there's not much research when it comes to looking at beetle-Shannon diversity and how it is related to ecosystem function. And we think that it's because beetle-Shannon diversity has a lot to do with soil diversity, soil respiration, which is a large part of ecosystem respiration, and that is where some of the difficulty in predicting ecosystem respiration comes from. And then we saw that including these metrics improve the predictions of our models. And lastly, the polynomial regression model outperformed random forest models and linear models for predicting ecosystem respiration and net primary productivity. Now we think that for the polynomial regression model, if we looked at the bivariate plots, we saw that the relationship was almost linear. So the random forest might have been overestimating or capturing some complexity there that was not as necessary, while in the polynomial regression, we were able to manipulate mean annual precipitation and have the other two variables be linear. So with that, I would love to thank my co-authors, Kasum Naitani, who was my doctoral advisor, and then Dr. Jingfeng Zhao, who provided the eddy covariance data from the University of New Hampshire, and the EAGER grant who supported this research, and NEON.
Yeah, thank you, Anna. So now, floor is open for question answer session. So if you have any question or comments, anyone, please. Thank you.
Thanks for your talk. It was really interesting. It's interesting to see the strength of the relationship with beetle diversity, especially in terms of having more predictive capacity than plant diversity, say. And I think your interpretation of could that be a proxy for something else or linked to something else like soil conditions or soil moisture. So is interesting. Would your takeaway be that, because if that's true, then why not just use soil data to improve predictions of ecosystem respiration and NPP? But that data can be hard to come by and can be hugely heterogeneous from one location to the other. So are you suggesting that these kinds of other diversity data should be more interrogated and then potentially used as proxies for other information that's hard to measure? Or do you think there's relationships, more direct relationships between, say, beetle diversity that are worth better understanding themselves? Did that make sense?
Yes. I think in terms of beetle diversity, I think a lot more work needs to be done on their role in relations to ecosystem functions, because it was really hard to find papers that actually explicitly look at that. So I think that's an important place to look at. And also, I feel like beetle diversity, science has shown that beetle diversity could also be a proxy for ecosystem health. So did I answer your question, sort of? Okay.
Yeah, thank you. Any other question or comments? Yeah, If there is no question or comment, thank you, Ayana, once more. And this is the end of this session. So I would like to thank all the participants as well as the speaker for joining this session. Thank you very much. Thanks a lot.